Welcome everyone. This is Talk to Toe back again with broke grad students because that's who we are. Joined today by Andrew Phan. How you doing, Andrew?
Hey, good, Chris. Hey, how are you doing?
Doing well, man. Well, Andrew and I have known each other for well, I guess it's been like almost 3 years now.
Yeah, that's kind of nuts. Pretty much
Yeah,
whenever we started grad school.
yeah, that's crazy. So Andrew, you're also a PhD student at UC Irvine, right?
Yep.
And whose
Yeah.
lab are you working in, and what are you doing there?
So I work with Dr. Tim Downing, and we work with stem cell reprogramming, and it's really cool.
Cool, cool. So maybe we can start with how you ended up in Downing's lab.
Yeah.
Tell me about that story because I know a little bit of it, but obviously it's been a while. So
Right, yeah, definitely. So I started off as a master's student. I graduated from the University of Kentucky and with my bachelor's in science in biosystems and agricultural engineering, which is basically biomedical engineering plus tractors. So I, I was like, might as well try to go get my master's in biomedical engineering. So maybe I can get like a job in industry or, just be more educated in general. So I applied and I got into UCI, which for whatever reason I really loved the idea of going to UCI. I had been there I think as a kid, but that was pretty much my only exposure to it. But for whatever reason, in my head, I always wanted to go there. So I made it, and then I started as a master's student, and I really enjoyed my work and my time and my lab mates. So I decided, hey, why not just stay like 3 more years and do my PhD? So that's pretty much how I ended up into the PhD program, and Dr. Downing was cool with it. And yeah, luckily I got some funding through the local department, and good to go.
nice. So did you grew up in California, right?
So actually, I was born in Long Beach, and I lived there until I was like 5. But then I moved to Kentucky when I was 5, and I lived there ever since until I finished my bachelor's degree.
Okay.
Yeah, but I would visit in the summers because my family is here. My mom's side, at least, of the family is here. So we'd visit in the summers and go to the beach or go to Legoland or all that fun stuff. And
Yeah,
I
like
always there's so much to do here. It's really great, and the weather's always nice.
yeah, yeah. What's the weather like in Kentucky?
So the weather is, I would say, bipolar in Kentucky. Basically, one day it could be like snowing and really cold, like minus like 0 degrees, and then the next day it could be like 6 degrees, warm, sunny, like really nice. Um, so yeah, the weather in Kentucky, we have all 4 seasons for sure, but it changes pretty frequently.
Nice.
um,
Well, I
yeah.
mean, yeah, that's most parts of the country. California is an oddity, I feel like.
Yeah, definitely. California is like super nice because it's always like sunny. The coldest it gets is like 50, 60 degrees, and
Yeah.
I don't know. It's so steady.
Okay,
um,
so when you were uh I guess choosing labs and stuff, uh how'd you end up with Dr. Downing?
yeah, so I looked at the website for UCI and just like at all the professors and what they did, and I knew that I wanted to do something more biologically related just because I had done some cell culture work at the end of my undergrad. I thought it was like super cool. You're working with cells and like they're living and you're taking care of them and they can give you information. I thought that was so cool. So I looked at labs that were more biologically based, and I saw Dr. Downing was working with epigenetics and stem cells. I was like, I have no idea what epigenetics are, but I've heard of stem cells. I think they're super cool. And I looked into it more, and just the concept of working with stem cells and epigenetics, I thought was just it was just too cool to like pass up on. So I asked if I could join his lab, and I was like, yeah, my last year at UK, I would just email Dr. Downey like, hey, can I join your lab? I also emailed a couple other people. I didn't hear from them, so luckily Dr. Downey responded, and he was like, yeah, why don't you come check out my lab in the summer? So I did that, and funny story. So I was driving from Alhambra to Irvine because that's where my family is, and I told my GPS like, hey, don't take me on toll roads. So I got onto the 73, which is a toll road, but it's only a toll road after you pass UCI. But I didn't know that. So whenever I saw that sign, I got off the freeway and I was like, what's going on? And so I just took me like a long time actually to get to UCI. But yeah, I like visited the lab. Everyone was friendly, and Dr. Downing was really cool. So I worked in his lab during the summer as basically a volunteer, and I got my feet wet, right? And I enjoyed cell culture, and I enjoyed just the whole experience in his lab. So I matched with him essentially that summer before I even started.
Nice.
um,
Yeah. So for the people who are like listening who like aren't really familiar with what epigenetics is, like, what's your summary? And it's a very broad topic, obviously, but what's your summary of what epigenetics is, and how does it relate to stem cells and what you guys are doing?
right. So everyone has essentially the same genome, right? Their genes are very very similar, but we're very different from each other. And the reason why that is because your genes and its expression is regulated by something outside of your genome, and that's what we call the epigenome. Like it's meta, it's epi, it's one step above your genes. And basically, what that means is that your genome, your genes can be methylated or they can have acetylation marks. And essentially, what those are is you add a methyl group to your DNA, and what that typically does, not in all cases, but typically does, is it reduces your expression of that gene, which is important in whether or not your your cell does a certain action or not. And then acetylation is, in general, the opposite, where it opens up the chromosome and it allows the gene to be transcribed more. So, uh, yeah, that's how I would generally describe epigenetics. There's so much that we're learning about every day, and that
Mhm.
those two rules are very not true. But that's how I would start by
Right.
explaining.
I mean, it is also a newer area of study. I think that's important to note, right? Because like I feel like Dr. Downing was like one of the main like people in
Hmm.
the field, right?
Yeah, he is a pretty big deal, I would say, just in terms of he has like a nature paper, which is huge, which is a really popular journal. But he was one of the first people to discover that biophysical cues are able to change your cell behavior and your epigenetics specifically in that way. Yeah, I guess to be more specific, basically what he showed was that if you take a stem cell and you put it on these microgrooves, which are just, if you want to think about it, it's like a stripe pattern on the surface of your cell culture um, petri dish, you can enhance the ability for that stem cell to uh, be a stem cell. It's basically more pluripotent, and um, that was controlled through epigenetic modifications that he found that correlated with that change. Um, yeah.
Yeah, that's pretty crazy. Yeah, I think cell work has always been interesting. It's like on one hand, it's cool because you can kind of see what's happening in like something that's living.
Hmm.
But on the other hand, it can be incredibly frustrating sometimes because you
Yeah,
can't always
definitely.
exactly control what's going on. So
Yeah, definitely. I agree. Especially with stem cells, right? They're so fickle. Like they never want to do what you want to do. So you're doing your experiments like over and over and over until you figure out basically how to work with stem cells in general. Just because, yeah, if you do one thing wrong, like they'll just they'll turn into a hair cell or an eye cell or like something else that you don't want them to be.
so when you guys are working with stem cells specifically, like where do you guys usually get them?
So I specifically work with this this human fibroblast cell line, so skin cells from humans, and they are special because they have the ability to turn into stem cells just by adding doxycycline, which is just like an antibiotic, into the media, and that activates a cassette that that activates the four Yamanaka factors: OCT4, SOX2, KLF, and c-Myc. And those are what you need to turn a skin cell into a stem cell. So I work with human skin cells and I turned them into induced pluripotent stem cells.
Got it. Yeah, that was also a huge groundbreaking discovery from like Japan, right?
Right, exactly. Yeah, Dr. Yamanaka from Japan, he found out that you can take a human skin cell and turn it into a stem cell, which is a big deal because normally when you're a baby, right, you have stem cells and those are able to differentiate or turn into any other cell in your body. So all your cells in your body were made up from this undifferentiated stem cell. And that has really huge like benefits if you want to do anything in say regenerative medicine, like you want to regrow an arm or a heart or brain or something that normally you can't regenerate. That's why people are really interested in stem cells, and I thought that was like a really cool application and something I wanted to study.
Yeah, that's also like how, I mean, I think the reason that they were studying that was because of all the like ethical issues that the Bush administration had like put a moratorium or something on like stem cell research for a couple of years. So then they were like trying to find ways to get stem cells like in other means or whatever.
Yeah, definitely. This is a much more ethical way to approach stem cells because normally people think you just take them from babies or fetuses or whatever, you take it from a potentially unethical source. But this way, there is not unethical because you can just take a skin cell from a consenting adult, and then you can turn that into stem cells that would be able to turn into whatever. You get the full therapeutic effects without having to worry about the ethics as much.
Nice, that's cool. Yeah. So, what is your research looking at right now specifically? I think you kind of mentioned it briefly, but I know everyone does something different.
So we found out that cell adhesion is really important to the process of reprogramming. So in general, turning a skin cell into a stem cell is super inefficient. Like normal ways, you have less than 1% efficiency. So if you take 100 to 1,000 skin cells, you would maybe get like one stem cell out of all those if you activate this OSKM cassette. So that's like really inefficient, not great in general. So what we did is we looked at cell adhesion and we thought maybe cell adhesion is not very good for, it basically works against this process of turning a skin cell into a stem cell. And the reason why we thought that is because there's this paper by Adam Engler, and he looked at how stem cells were sensitive to the physical surroundings. So if you put a stem cell on a soft substrate, kind of like the softness of a brain, it would turn into a neuronal cell. If you put it onto a hard substrate like bone, it would turn into like a bone cell, an osteoblast. So we know that stem cells they're sensitive to their physical surroundings. So we thought nobody's really looked at specifically cell adhesion. So we looked at how cells feel their surroundings. So cells that have, in general, these two receptors. So they have on the bottom, they have these integrins, which allow the cell to attach and to feel the substrate underneath it, the usually extracellular matrix or ECS. And then they also have these receptors on their sides called cadherins, which allow the cells to attach to other cells, its neighboring cells, and also kind of talk to them in a way. So these cells have ways to feel their physical environment and their neighbors. And we thought, what if we perturbed that? What if we prevented them from being able to do that? Because it seems like those signals could tell it to turn into, say, a brain cell or a skin cell or what have you. So if we stop that cell from being able to feel those surroundings, we can potentially increase the efficiency of reprogramming. And that's what we actually found, which is super cool. So we knocked down the genes related to cell adhesion, and we found an increase in reprogramming efficiency from turning a skin cell into a stem cell.
Was that like a tenfold increase, or was it like how big of an increase was it?
So we found up to a 27-fold increase. So it's pretty significant, I would say. And what was cool, so we tested out 103 of these genes, and I would say definitely over 80% of them increase reprogramming efficiency compared to the average of the controls. So that was a pretty big deal, I would say.
Yeah, it's huge. Yeah.
Yeah. Additionally, what we're looking at, there in cancer, there's these cells. Whenever you do chemotherapy, right? Sometimes people relapse, and one of the theories right now is that it's because of these like drug-resistant cancer cells. They call them cancer stem cells, and cancer stem cells are important because they are more resistant to drugs, but they also are able to turn into more differentiated forms of cancer cells, and they're able to repopulate the whole population of cancer. So basically, if you leave one of these cancer stem cells alive after chemotherapy, it will give you cancer again, just by spreading all over again. So what we did is we looked at maybe how does cell adhesion in this context play a role in turning a normal cancer cell, like a differentiated cancer cell, into, say, a cancer stem cell. So basically, we again knocked down a cell adhesion gene in these human lung cancer cells, and what we saw was there was an increase in cancer stemness. So it seems like maybe there's a link between cell adhesion and just being able to gain pluripotency in general and in different contexts, not just stem cell reprogramming.
Have you guys ever looked at, cell density and like how, like, cause you mentioned that, cohesions increase are used, I guess, by cells to communicate with each other. But I'm wondering, like, is the density of the cells population that you're culturing in, and does that affect like pluripotency or like stemness?
Yeah, definitely. That's a really important factor, I would say. So there's this interesting, I don't know if I would trust this study, but there's a study that that they showed where if you pattern the extracellular matrix into like islands, into small islands, the cells will not have a lot of area to stick to because cells stick to ECM, right? So they will be all balled up and kind of like almost like a sphere because they're barely attached to the actual floor. And they found that by making a specific size and a specific density of cells, you're able to turn a mouse embryonic fibroblast into a mouse induced pluripotent stem cell. So I would say density is definitely something important, and you can play with that by playing with the area in which a cell can adhere to its environment.
Yeah.
So.
Yeah, I mean, cells are definitely interesting. I mean, the only cells that I've ever worked with are, I worked with HeLa cells, but that was just learning how to culture cells. And then I've worked with astrocytes and neurons.
Right.
Yeah, and it's interesting because like, because astrocytes are kind of like immune response cells for the brain, but they naturally exhibit that clumping. Because like that's what they do. So like if you have like damaged neurons in an area, typically any cells that are astrocyte related cells will kind of aggregate to that area as like a kind of like a defense mechanism, I guess.
Yeah, that's really interesting. Yeah, certain cells just clump up, and yeah, it's part of their biological function. Yeah, how was your experience with cells, I guess?
It was fun. Yeah. I mean, I had a very small experience because I've always mostly worked on the protein and like electrical side of things. So, yeah, it was really just for my like senior design project undergrad. We were trying to see if we could reproduce a platform to create like mild traumatic brain injury. So like, I guess the biggest thing for brain injury is like we kind of all know what concussions are, but there's like this kind of gap where we don't know what happens if you get like a lot of small hits over time. So especially for like NFL players, like they might not get a concussion every single time, but they're getting hit every single time. And like your brain is still moving within your skull ever so
Yeah.
slightly.
Hmm.
And a lot of that force is actually the fluid moving against your brain.
Yeah.
Where a concussion is like your brain actually like hits your skull,
Hmm.
but there's like the times where your brain doesn't hit the skull, but like it's still being sheared by like the fluid movement. So we were trying to find a way to like create a platform where we could study that. It's really difficult to do because it's like
Yeah.
you have to like control the flow of liquid over cells,
Uh-huh.
which is yeah, kind of difficult. But there's
Definitely.
like a lot of studies showing that like maybe that over time, if you have a lot of this sheared like fluid flow over cells, it
Yeah.
can affect things kind of like similar to what you're talking about of like cell adhesion or like cell interactions with each other where you have these like micro tears over time.
Yeah.
And eventually that can
Definitely.
lead to the issues that we see as like, CTE or like, those really long-term mood disorders that a lot of NFL players have.
Right.
Um,
Mhm. Yeah, that's super cool. Yeah. It's always really hard to model a real life system with cells just because everything, yeah, you do your best to make sure you can control everything and make sure it's as similar, but cells always have different ideas.
yeah,
um,
definitely.
but yeah, that's super cool. Let's see, so I actually have, I was wondering if I could take over the show as the host for 1 second.
Oh yeah, go for it.
So I was thinking about the name of the podcast, like Talk to the Toe, and I was like, oh, that's really cool because alliteration, and that had me thinking. I don't know. This is how I think. I thought like, what if it had a different name, or what if you had names for different segments and stuff like that. So I came up with a list of alternative names that you could use for your podcast, free of charge. This is a free service as a friend and as a fan of the show.
All right, let's hear them.
So, okay, yeah. So this is kind of similar to a show. I don't know if you've ever seen like Nathan For You, but he's like a businessman, and he essentially goes to these businesses and comes up with like ridiculous ideas to save them. So an example is like there's this TV place actually in uh, Alhambra or Pasadena. I actually passed by it before, and it was having trouble competing against Best Buy. So what he told them to do is take advantage of Best Buy's price matching, their price matching. So he lowered his prices for TVs, and he had a bunch of people go try to buy the TVs at Best Buy at this really low price that he was selling it at. And then whenever people would come into his store, they would have to. He had a dress code, so they had to be like in a tux or like a really fancy, like this dress. And then they would have to crawl into this tiny door, and behind that door was a live alligator. And behind the live alligator was the actual TV on sale. So he comes with just like ridiculous ideas like that, and I think it's super entertaining.
Oh my gosh.
So this is kind of like on that level of absurdity.
Okay, okay.
Okay, so here's the first one. So the first one is Socket Tomy. So all these are pretty much going to be puns on your name. It's going to be great. So Socket Tomy, going toe to toe with Chris Toe. So and then there's the christening.
Oh my.
Next one is the topography of science with Chris Toe. And then this one's a personal favorite of mine. What in Toe Nation, y'all? And then this one's based on a song by the Zach Brown Band. So toes in the water, toes in the sand. Not a worry in my, not a worry in the world. Cold beer in my hand.
Your
So
Kentucky's really showing right now.
it's just what I do. It's part of my blood now. The next one is this one's actually based on Amanda Bynes, like her skit show, Totally Amanda or whatever, in from a long time ago. So it's Totally Chris instead of Totally Kyle. And then
I remember
there's
that. It's like 90s.
yeah, right, exactly. I thought of that the other day. I was like, yeah, we could totally use that. um, yeah, so then there's Chris Topher. I hardly even know her. Next one is there's always tomorrow. Um, and then so slowly, I thought about this like 2 nights ago at like 3 a.m. So I was kind of getting hungry. So I thought, what about spicy tofu? Or okay, so here's another one. The toe down. I think there's like a show on ESPN called like the lowdown or something like that.
Yeah, I actually write for that show.
Oh, there you go. Yeah, perfect. Yeah, so there you go. You could totally like spin off of them, do something really cool. And then this is one. It's like if you want to do like a Disney podcast, it's called Christopher Robin. So yeah, that could totally be a great Disney podcast on Star Wars or whatever. And this next one is like it's like something like maybe a mom would say, it'd be like eat your tofu.
We gotta start doing some recurring segments or something.
Oh, for sure. That's not even all of them. So do you watch Netflix? This is actually one of my favorites too. Have you ever seen Netflix? Okay, so this one is like based off one of the super popular shows during this pandemic. It's Toger King instead of Tiger King.
Oh my God.
Just think about it. I mean, like, you don't have to use all of them, but you should. That kind of deal. And then here's another good one based off of like a really classic anime movie. It's My Friend Toto Chris.
Oh, yeah.
I can keep going, but like those are some of my best ones that I thought of. I don't know,
spent
what
a
do
lot
you
of
think?
time thinking about it.
Yeah, I mean, long-time fan, first-time caller, so I gotta make sure I'm prepared.
Very nice. I'll need you to send that list over because that was a lot of it.
Oh, for sure, yeah. Yeah, don't worry, I'll be texting you this instant this is over.
Nice. Yeah, I definitely want to do like a group one of these at some point with like a couple of the BME guys because I've been doing mostly one-on-ones, but it'd be interesting to get like just a bunch of grad students in a room and just be like, all right, let's just talk about whatever, see what ideas come up. But
yeah, definitely. I think the hard part about being in PhD, right, is like the first year you have a little bit of time because you're just figuring stuff out and you don't really have a solid project yet usually. And you can spend time with everyone in the cohort, right? You can just like hang out, play volleyball, play soccer, go to parties, whatever. You can actually do things, right? But after the first year, like the science really kicks up, right? And your classes and everything, you're just like, you literally have no time to hang out. So this is, I think, a really great, this podcast is a great service in like allowing us to reconnect and like, just talk about our experiences because I miss like talking to everyone. I miss hanging out.
yeah.
All that good stuff.
Yeah, I mean, it's like nowadays, it's like maybe I see people in passing in the hall.
Right, exactly.
That's only if they work in your building, too.
Yeah. Campus is usually a big place, so
Yeah.
running into people in the hallway is not always very, very certain or very common.
Yeah, I think it's like definitely. It's like I think like half the people in our year, I have no idea what stage they're at in their studies or anything like that. And it's just like,
right.
We don't really go to seminars anymore unless they pertain to our research,
Yeah, exactly. Yeah. Yeah, only if they're useful for us anymore.
Yeah. So I don't know. We'll have to figure out some time to get like a couple people in here. If you know of people who are interested in doing something like this, within your lab too. Like I'm always looking for people to talk to, interview, just get to know them more,
Yeah, definitely. Yeah, I think I can maybe line up some cool peeps. I'll let you know.
Yeah, we can do a group thing or something too.
Yeah, that'd be super fun. Honestly, like it'd be kind of interesting, right, to do a podcast like or like an interview with everyone from a lab, right? Or like at least a bunch of people from the same lab and just to see like how the different labs are so different.
Mm-hmm.
Yeah, just the different personalities or whatever. But yeah, I don't know. Just, yeah, I think that'd be a really good idea.
Yeah. I think it's doing it virtually is actually probably more feasible in that way because when I first started this, I was doing it in person, right? So that was like that was good, but it was also kind of difficult to schedule because like people had things they need to do and like
right.
I'm just inviting people over to my apartment to hang out for a
Yeah.
minute.
Yeah, well, I guess what we should have probably prefaced the show with is that right now there's a global pandemic that's happening, which is why no one can leave their house, and it's yeah, it's serious. So I don't know how's everything with you? You know, you and your family and friends and all that.
Yeah, I mean, my family's doing well. My brother is trying to graduate from
Hmm.
undergrad right now, so they're not
Is
having,
he
their graduation ceremonies as is like
right?
most universities.
Yeah. How's he feel about that?
I think everyone's like a little bit bummed, I think he's bummed too, but he's also
Right.
glad to be done,
Uh-huh. Right. Right.
as is most people by the time they hit senior year.
Uh-huh.
Um,
Yeah. Definitely.
yeah, I mean, my fiance is working in a hospital. She's a nurse.
Hmm.
Um, yeah, I've been like I've only been going to see her like when we feel like it's like safe. But like so far, she hasn't had any patients. But she just texted me early this morning that she has COVID patients,
Oh, gotcha.
which
Gotcha.
is like the
Yeah.
first COVID patient she's had since this has started.
Okay. That's
Um,
good.
which is a good sign. So we're just trying to figure out like what is like the safe thing to do, the responsible thing to do, and like whether or
Yeah.
not like we should see each other for a bit.
Uh-huh.
It's like we're trying to get married in September,
Right. Yeah.
so it's like
Uh-huh. You want
at
to
a
spend
certain
time
point,
together?
like if she's gonna get it, I would have gotten it anyways. So like, uh,
Yeah. Uh-huh.
but like right now, we live with roommates and stuff, so we're just trying to be respectful of them also.
Right. Right. Yeah.
Yeah, it's weird navigating that stuff. But everyone's healthy still.
Mhm.
Around here, like we've been pretty lucky from what I've heard, at least from what I've heard from her, and then other people in like the medical community. Like Southern California is like we have cases for sure,
Yeah.
um, but like the hospitals are not necessarily overloaded or anything,
Hmm.
which
Hmm.
is like what we were trying to prevent. So that's good.
Yeah, definitely. It's kind of crazy because like I feel I think in the news, right, the OC got some of the first cases of COVID-19 in America, at least that we identified.
Hmm.
Um, yeah, but it doesn't seem like it is as bad as it could be, which is a good thing, right? Because we have the stay-at-home order and stuff like that.
Yeah,
Um,
yeah.
yeah,
So I don't know. At a certain point, like you kind of have to weigh the benefit of like letting people go back to work and like
yeah.
trying to keep like because the thing is like everyone's gonna get it at some point. It's just like a
Hmm.
matter of like uh what's the cost of that, and like if everyone's waiting for a vaccine, like
Hmm.
if you don't have economy to make the vaccine, then you're kind of in a dilemma there too. So uh,
Yeah, that's an interesting point. That's kind of like what the people, the protesters in Michigan are doing, are like protesting for.
yeah.
Um, yeah, I think it's a really interesting issue. Yeah, how do you weigh the value of saving lives versus like the tangible loss of like businesses and like the welfare of people, right?
Yeah.
Um, yeah. So actually, in this class I'm TAing for, it's a professional writing class, and one of the things that we talk about is ethics and different ethical frameworks. So something that people typically use but don't really think about is this framework called uh, act utilitarianism. And basically, what that means is that you think about the value of things of doing things on one way, and then you think about the value of doing things on the other way. Typically, you quantify it with money, but like the problem with that is that um, how do you quantify the value of a human life, right? How do you
Hmm.
value um, the the loss of a business, because those have profound effects, and it's really tough to calculate. But it's like whichever way it does the least harm or whichever way it does the most good is typically what people want to go with.
Right.
Yeah.
Yeah. It's difficult because it's like at the same time, like um, I think there is some issues with like how public health is like communicated to the general public because
Definitely.
a lot of people get really upset with you when you're like wearing a mask and
Hmm.
you're like less than 6 feet away from them.
Right.
Or like vice versa, where you're not wearing a mask, but you're like 10 feet away from someone, like they're still mad at you because you're not wearing a mask.
Uh-huh.
But
Right.
it's like on a public health level, if you study public health, you know that like having one or the other is like kind of like if you have a mask, like you're okay to be like a little bit closer.
Right.
If you don't have a mask but you're farther away, like it's still fine. Um,
Right. Right. Yeah.
but everyone conflates it that you have to do like both of them, like no matter
hmm
what.
hmm
Um,
Yeah, I guess just to be double safe.
yeah, just to be double safe, I guess. I don't know, but that I think
um,
that creates a lot of paranoia within
yeah,
people, and like that in itself might not be a positive thing.
I
So,
agree. Yeah, it caused a lot of like maybe unnecessary stress.
yeah,
Um, I know that honestly, when people started first like wearing masks, it's like I don't want to wear a stupid mask. Like, why did I want to do that? I live in America, like I live in land of the free and
yeah,
whatever. But yeah, it's important to think about the people that don't have that luxury to be like, oh well, if I get it, I'll probably be okay, right? So
yeah.
yeah, like is it ethical to just because it's convenient for you, is it ethical to not practice safety concerns as much as you can reasonably?
Yeah, I think a lot of people have been talking about because what they did in Sweden was like they basically told all the people who are older to shelter in place.
Right.
But they like anyone who's like still young, they're like just keep working and like hopefully everyone gets the virus and
hmm
like gets herd immunity that way faster.
Exactly. Yeah.
Um, so it would be interesting to see in a couple of months if that like ends up working better because they might if they do that and like it works and they might not get a second wave.
hmm hmm
Like we might get a second wave because we're all sheltering in place, but
Yeah.
they might not get a second wave because everyone would have already gotten it. So
But the problem with that, right? We don't know whether or not you can be reinfected with the virus or
yeah,
not. So that's assuming that you can't be. Yeah, your antibodies that you develop from obtaining the virus will keep you safe.
mm,
So it's still a risky-ish strategy. It's I would say it's likely that you would have some sort of immunity after you catch it, but you never know. Um, and it's kind of funny because what Britain tried that, United Kingdom tried that, and then Boris Johnson's like, okay, never mind, maybe we shouldn't do that. And then he ended up getting coronavirus, and it's just
yeah,
so yeah, it's just crazy. So in general, I don't know if I would say the most ethical thing is just like, oh, just infect everyone, it'll be
yeah,
okay. um, yeah, but I don't know. Yeah, it's interesting. There are a lot of pros and cons that have happened because of this virus, right? I don't think things are going to return back to normal after all this is said and done, because everyone's working from home. Dude, the traffic in LA is like non-existent. It's fantastic, um, but it's yeah, you still have to be. You're much more weary, I guess, of people around you though too, right? Because you're always thinking like, what if they have it, or what if
mm,
what if they're not being safe, then it would affect me, and even though I'm trying to be safe.
yeah. There's definitely an interesting social element to it too. Like, I mean, I've joked with this with other people. It's like, what are we like? What's gonna happen to like the kids and like who are growing up right now? Like,
yeah.
obviously, this is gonna severely affect like how they view interacting with people.
Yeah.
Um, and I'm like talking about like younger kids, like if you're in high
hmm
school, you kind of know what's going on, but like,
Yeah.
if you're like 5 or 6, like you're not going to understand necessarily why you can't like hug your friend or like see your friend, you're
Right.
going to school.
Exactly. Definitely. Yeah. Um, that is super interesting. And honestly, I would say for like middle schooler and high schoolers, I think it's really tough, right? Because uh, even in summer, right? In summer break, I would get tired of summer break because I don't get to see my friends nearly as much. And this is like that on a like a macro scale. Like nobody gets to see anyone, even outside of school. Um, and you're really developing, those social ties, and you're learning how to learn. You're learning how to socialize. You're learning all these really important skills that you'll use later in life, right? Um, so I think it's pretty tough for them just because as adults, typically you have more established relationships that you can lean on and things like that. But but younger adults and younger students, like they don't have that yet. So this is going to be really difficult and really isolating.
Mhm. Yeah,
Um,
I think as an adult, like you've also learned more how to build those relationships,
yeah,
regardless of the circumstance. So like, even though it is more difficult now as an adult, typically adults have more of that experience to know how to continue doing that, just in a
yeah,
weirder way, like.
definitely. Um, yeah, it's kind of crazy how I don't know. You don't think about these things, but you can learn how to do a lot of different things. Like you can learn how to make friends better. You can learn how to you can learn how to do cell culture. You can learn how to do like so many different things if you think about it. None of these things are you're born with, right? And that's I think that's a really cool way to look at things. That you can learn these skills rather than I was not born with like this or that. And uh, yeah.
Nice. Well, we've been talking for quite a while now, so.
Wow. Yeah. Oh yeah. It is already 11.
Yeah, don't want to keep you too long, but yeah, maybe the one last question is what are you looking forward to in the rest of the year? I know it's a little bit hard to know for sure, but what is your plan? How far out do you think you are from graduating?
That's a great question. That's $1 million question. So I'm starting my 4th year now. Wow. So yeah, my 4th year at UCI. So I'm hoping to graduate by the end of my 5th year. So like I'll have this year and then next year, next school year. So that's my plan. We'll see what my experiments and what my cells tell me, but I'm hoping that. Um, but I'm looking forward to my girlfriend she's in San Francisco and I'm in LA, so we're long distance. So I'm looking forward to just spending time with her again. Yeah, it's tough doing a long distance relationship, and then on top of this, I can't even go visit her if I wanted to, kind of deal. Makes it more difficult. But yeah, I mean, I'm also looking forward to going back into lab. Maybe not necessarily to do the work, but to spend time with my lab mates because like we get along really well. We do a lot of fun things that may or may not be related to lab work, in lab. So it's always good to reconnect with them, and I think it's fun to bounce ideas off too because you miss that whenever you're not with people. Just
Yeah.
like yeah, getting feedback and having their ideas and input. Um, yeah, so I think those are some things I'm looking forward to. What about you?
Yeah, I mean, I'm looking forward to getting married, hopefully. So
Definitely, yeah.
if that happens, well, yeah. I mean, our plan is we're gonna get married no matter what. So
Hmm. Hmm.
if we can't have a ceremony, we'll have a ceremony. But
Yeah,
if not,
gotcha.
like, it's not the end of the world. And like,
Hopefully not.
yeah, hopefully. I say that now, but I can't see the future.
Yeah.
Um,
Just kidding. Yeah, I think it should be better by then.
yeah.
But
I
yeah,
mean, I'm
that's
looking
it.
forward to our collaboration too with our
Yeah,
labs.
definitely. I'm super looking forward to this because you guys do all this really great computational work and you guys like a bunch of nerds and are awesome. And I really I'm looking forward to like seeing how we can combine, like biology plus like all this data mining and machine learning and all these great buzzwords.
Yeah, I mean, we're all nerds, I guess. So
Yeah, very true.
yeah,
um, yeah, definitely. I am really looking forward to that. I'm glad that our PIs are getting along and that we can do collaborations like this. That's I think one of the strengths of UCI is like professors are willing to collaborate.
yeah. That was part of the reason why I really liked UC Irvine was like I think the level of collaboration here between professors is actually pretty unique.
Hmm.
Um, because like not all universities are like that. It's like
Hmm.
especially within the same like university.
Yeah, definitely. It's some people just view it as like a zero sum game where like if someone gets resources, I will never get resources, or like that's off the table. And yeah, it's not very productive in academia, but that's just the way it is. It's like a game.
Yeah, unfortunately, when things are all based off funding and grants, like
Right.
that's kind of the drawback of having a funding system like that. But there's not. I don't know if there's a better way to do it, really.
Right, exactly. Yeah, it's such a hard problem to solve, but yeah. Um, I don't know. Hopefully someone's working on that. I don't know.
Who knows?
Yeah.
All right.
Um,
Well, where can people find your work?
my work. We actually don't even have a lab website, so
Oh, really?
yeah. So what? I don't know. Just hit me up in real life. I think that's the best way to do it.
Well, if you
Yes.
guys need help setting up a lab website, I can help you with that.
Okay, yeah, I might take you up on that. We'll see
Yeah.
what happens. We got really close. Like we have pretty much everything on there. I guess we just never really published it. I don't know.
I need to tell Dr. Brody that we need to update our lab website too, because it's like it's from the 90s. So
That's right. It's pretty dope. It takes you back.
it's literally just a list of publications.
Right, exactly. I went on there. It's like, oh wait, like is this just one of the pages like on this website? I was like, no, this
Yeah,
is
yeah.
it. Um, yeah, super informative. I would say that. Straight to the point, everything you need. um, but yeah, yeah. So I'm not a big like social media person or anything like that. So I'm not really on anything. You can find me on Animal Crossing. That's where you can find me right now. I've been playing a
Yeah.
ton of Animal Crossing. So yeah, find me on there.
All right.
um,
Great.
yeah, thanks so much for doing this, Chris. This is really fun.
Yeah. Thanks for coming on. It was a pleasure.
Yeah, definitely. I'll see you around, Chris. Take it easy.
Be sure to like and subscribe to Talks with Toe on Spotify, and also give us that 5-star rating on iTunes. Also, be sure to go to YouTube and subscribe to Talks with Toe. Talks with Toe is written and produced by Chris Toe.